Every heartbeat, every thought, every movement of your body depends on tiny molecular gates embedded in your cells. These gates, called ion channels, control the flow of electrically charged particles across cell membranes. For decades, scientists knew these channels existed and understood their importance, but their exact structure remained one of biology's most elusive mysteries. Roderick MacKinnon, an American biochemist and molecular biophysicist, dedicated his career to unveiling this hidden architecture of life. His groundbreaking work would transform our understanding of how cells communicate and function at the most fundamental level. Ion channels are proteins that span cell membranes, creating pathways for ions like sodium, potassium, calcium, and chloride to pass through. These channels are remarkably selective, allowing only specific ions to cross while blocking others that are nearly identical in size and charge. This selectivity is crucial because the controlled movement of ions generates electrical signals that power everything from nerve impulses to muscle contractions. Before MacKinnon's work, scientists could measure the electrical activity of ion channels and predict their behavior, but they couldn't see their actual structure. It was like knowing a lock works perfectly, but never seeing the shape of the key or the mechanism inside. MacKinnon approached this challenge with a technique called X-ray crystallography. This method involves crystallizing proteins and then bombarding them with X-rays to reveal their atomic structure. The difficulty was that membrane proteins like ion channels are notoriously hard to crystallize because they naturally exist within the oily environment of cell membranes. In 1998, MacKinnon and his team achieved a breakthrough. They successfully determined the three-dimensional structure of a potassium channel from bacteria. For the first time, scientists could see exactly how these molecular machines were built, atom by atom. The structure revealed an elegant solution to a profound puzzle. The channel forms a narrow pore through the membrane, and at its narrowest point sits a selectivity filter. This filter is lined with oxygen atoms arranged in a precise geometric pattern. Potassium ions, which normally wear a coat of water molecules, must shed this coat to pass through. The oxygen atoms in the filter replace the water molecules, but only for potassium ions. The filter's dimensions are too tight for larger ions and too loose to properly stabilize smaller ones like sodium. This discovery explained how a channel could distinguish between potassium and sodium ions, even though sodium is actually smaller. The answer lay not in size exclusion, but in the precise chemical environment of the selectivity filter, which was perfectly tuned to potassium's specific properties. MacKinnon didn't stop there. He went on to determine the structures of numerous other ion channels, including voltage-gated channels that open and close in response to electrical changes across the membrane. These structures revealed how channels sense voltage and undergo dramatic shape changes to open their gates. His work also illuminated the mechanisms of channel regulation. Some channels have additional domains that act as sensors, detecting signals like calcium ions or mechanical force. When these sensors are activated, they trigger conformational changes that open or close the channel pore. The implications of MacKinnon's discoveries extend far beyond basic science. Ion channels are targets for numerous drugs because their malfunction underlies many diseases. Mutations in ion channel genes cause conditions ranging from epilepsy and cardiac arrhythmias to cystic fibrosis and certain forms of deafness. Many existing medications, including local anesthetics, antiarrhythmic drugs, and treatments for epilepsy, work by modulating ion channel activity. Understanding the precise structure of these channels has opened new avenues for drug design. Pharmaceutical researchers can now see exactly where and how drugs bind to channels, enabling the development of more specific and effective therapies with fewer side effects. MacKinnon's contributions earned him the Nobel Prize in Chemistry in 2003, which he shared with Peter Agri. The Nobel Committee recognized that his structural revelations had solved a problem that had puzzled scientists for decades and provided a foundation for understanding a fundamental aspect of life. Beyond the specific structures he determined, MacKinnon's work established methodologies and approaches that continue to drive the field forward. His success in crystallizing membrane proteins inspired countless other researchers to tackle similarly challenging targets. The story of ion channel research illustrates how answering one fundamental question can illuminate entire landscapes of biology and medicine. By revealing the atomic architecture of these molecular gates, MacKinnon transformed abstract electrical measurements into tangible, visual understanding. His work reminds us that the most profound insights often come from seeing what was previously invisible, from making the hidden machinery of life visible to the human eye and mind. Today, researchers continue to build on MacKinnon's foundation, determining structures of new channels, understanding disease-causing mutations, and designing targeted therapies. The secrets he revealed have become the building blocks for the next generation of discoveries in cellular biology and medicine.